Supercoiling refers to the additional twisting of the DNA molecule beyond its natural double helix structure, resulting in a more compact and organized form of DNA that plays a vital role in several cellular processes such as transcription and replication. In other words, it can be defined as the process of supercoiling, which involves either overwinding or underwinding the DNA double helix to generate a twisted and coiled structure, aids in controlling and compacting the way that genetic information is packaged in cells. Supercoiling occurs during DNA replication and transcription when the DNA strands are unwound to expose the genetic information. The separation of the two strands leads to the generation of torsional stress, which can result in supercoiling as the DNA attempts to relieve this stress by twisting into a more compact form. Enzymes called topoisomerases help manage and regulate this supercoiling in cells. Supercoiling in DNA can be categorized into two main types, that is positive supercoiling and negative supercoiling. In positive supercoiling, the DNA double helix is overwound or twisted in the same direction as its natural helical structure. This means that the coils of the DNA double helix become tighter and more compact. Positive supercoiling is often found ahead of the replication fork during DNA replication and can also be important in regulating gene expression. Negative supercoiling, on the other hand, involves the DNA double helix being underwound or twisted in the opposite direction to its natural helical structure. In this case, the coils of the DNA double helix become less tight and more relaxed. Negative supercoiling is typically found behind the replication fork during DNA replication and is important for maintaining the overall stability of the DNA molecule.